Welcome to this podcast edition called Mendel and the Gene, the study of heredity. I hope that you'll get a lot out of watching this podcast. The main goal for this podcast is to, one, give a brief overview of Gregor Mendel's work that started the science of genetics. Number two, I will explain the basic details related to what the genetic material is made of and what gets passed from one generation to the other. And then lastly, I'll introduce the four main patterns of heredity that have been observed in nature. So when you see this picture of this gentleman writing on a chalkboard, he has various traits that we can observe. He's not bald, he's Caucasian, or at least seems to be. He's left-handed, and he's a male. As you're going through this study of genetics, you'll notice that it really is about the DNA in the cell's nucleus that is being expressed in visible ways, and so these two pictures are really contrasts in the large scale, which is the gentleman up the upper left, and then also the small scale, which is kind of a stylized picture of the DNA molecule. So as you're taking notes, I would strongly encourage you to take notes in the Cornell Notes fashion. You can see that the title at the top says Mendel and Gene. You also want to put the date there. And then remember at the very end, and even throughout the podcast, write down comments or questions that you'd like to go over with your instructor so that you can best understand this material. Some questions that inevitably come up in the study of genetics are myriad. Several of them are listed on the screen. The first is, how are traits passed from one generation to the other? That's really the fundamental driving question that Gregor Mendel had started with as he was trying to figure out how does the sort of the cycle of life or the continuity of life go from generation to generation. Second thing you may have wondered from time to time is, why do some members of your family have traits that are different than yours? And then why are there some that are similar? And then thirdly, why do some traits seem to skip a generation? So your grandparents may have one trait and your parents don't have that same trait, but then you have that same trait. So what's going on with there? How can we explain the skipping of those traits? So as we go through the study of genetics, there are usually some challenging words to overcome and to master as we understand what's going on with our DNA and with our chromosomes. So this word wall lists some of the more classic ones that may be very important for you to know. So a genotype is the two-letter code that we get from our parents. We get one of the two letters or pair from mom and the other from dad. That letter code that I was explaining in genotype is called an allele, and there are generally two forms of an allele. There's a dominant form and there's a recessive form, and the three ways that those two letters can pair up are listed as follows. So heterozygous is big A, little a, or it's a dominant and a recessive allele paired together. Homozygous recessive would be two recessive or small case letters that are paired together, and the homozygous dominant would be two dominant alleles or gene letters paired together. And then the phenotype is the physical expression of the genes. The expression is not always outward. A lot of times the biomolecules and enzymes and such that our bodies need are coded for by genes, but we never actually see them. But when we're ill, or when individuals suffer maybe a genetic disorder, you'll know by their absence how important they are. So as we go through this podcast, you'll see that I've grouped topics by related theme here. So first we'll look at Gregor Mendel and his work with the pea. It's become a classic story in science. Secondly, we'll look at the structural materials of what are genes made of, and what is that material that is passed from parent to offspring. And then we'll look at four hereditary patterns really briefly, so that you can be able to sort of pick out these patterns in various examples of nature. Four big ideas that I'd like for you to understand as we go through this. First of all, that science is really a progressive field. One individual will build on the work of predecessors, and this is no different than any other kind of field, I guess. But in this case, Gregor Mendel really was the first observational geneticist, where he really looked closely at the data and he tried to draw from that data trends or conclusions that would make sense for explaining the pattern of heredity. So he is the premier scientist that contributed to the advancement of genetic science. And what we know about science now, specifically genetics, is the result of many of his experiments that were then added to by other scientists after the fact. So no study of genetics would be complete without a brief understanding of who Gregor Mendel was. He is considered to be the father of genetics. He was very much scientifically minded. He studied physics and botany. He was a professor at a university. He was also very religious. He worked at a monastery, and among his many jobs was to tend to the garden, and that's where he first encountered the now famous pea plant. And it's from that humble origin that genetics has sprung up. So when he was doing his work, he was basically modifying some plants and doing artificial sort of cross-fertilization. And so this graphic just shows kind of the basic steps. So what he would do, or what anyone can do, is you take the boy parts and remove them. So the stamens are where the pollen or the sex cells are produced. So you cut those out of the purple plant, leaving the female parts behind. Since in these cases flowers are sort of bisexual. And then you would take another flower, in this case a white flower, and you would take a paintbrush and transfer some of the pollen from the white boy parts and then transfer it to the female parts. And so oftentimes you'll see the pollination happening in nature from organisms like bees, or even certain primates will do this. Lots of insects will do that. And so what he did is he just artificially crossed or fertilized the white plant and the purple plant. And then when that happens, sexual reproduction commences, and then you have baby peas made. And when those peas are planted in the ground, they grow up and they become adult peas. In this case, it's called the first filial generation. Filial means son, so it's sort of the first son generation from the parents. So that's basically what he was doing, but he did this for thousands and thousands of different variations of pea plants. So here, this is just basically showing that the honeybee has been replaced by the paintbrush. So this is a list of the different characteristics that he looked at. So there were 7 specific traits. You can see those listed on the screen. I think what's important to note here is, as you look at the F2 generation, you'll notice those are huge numbers. In each case, they are almost 1,000 F2 generation, or second filial generation. But you'll also notice that the ratios, when you do the math, it's usually around a 3-to-1 ratio, and that number is going to be important later as we look at the trends of heredity. So it was a lot of work that went into this, and he actually died before his manuscript was published and before anyone really understood the importance of what he was doing. So unfortunately, he never really got to get that pat on the back like he deserved. So this is the basic sense of what he did. So what he concluded was that in this case, if you have 2 flowers of different varieties, one purple, one white, both are coded for color, they just have different variations of color. What he noticed that in the parent generation, the white flowered parent was pure breeding, meaning that it was homozygous recessive, so it had 2 lowercase allele letters, and then the purple flower was true breeding, but it was dominant. And so you'll notice in F1 generation, you won't see any evidence phenotypically of the white flower. And then what he did is he took the F1 plants and either allowed them to self-pollinate, which is to just mate with themselves, or cross-pollinate. And you'll notice in that 3rd generation, the F2 generation, you'll have 3 of the purple flower plants to 1 of the white flower plants. So in essence of why do traits skip, in recessive combinations, in this case the white flower, the skipping is because the second allele in F1 generation is dominant, so it's heterozygous, and so one of those alleles is in effect overpowering the other allele, and so it's only when 2 recessive alleles get paired up together that they can actually express themselves. So they're always being masked or hidden by the dominant allele, and they're only revealed when they're paired up with their similar recessive partner. So this work with pea plants gave Mendel 2 insights into how traits pass from generation to generation. So the first law that he came up with is the law of segregation. So basically, two alleles are separate. So if you have a F2 hybrid, so it's a purple plant with a dominant allele and a recessive allele, that those alleles will separate when the sex cell is created. And so there's no sense that both alleles pass through the sex cells together. Remember that sex cells are haploid, so they only have one copy of every trait, not two copies, and so that's why the two alleles, they actually separate. So that's the first thing that he came up with. It's important for you to also understand that. Sex cells only contain one copy of every allele for every trait we have. So you can see that in here. So the P generation, they make sex cells. That's represented by the circles. And you'll notice that both of them are purebred or homozygous, and so they create sperm cells that are, or egg cells that are all alike. And that you'll notice in F1 generation, we have a heterozygote purple flower, and in half of the case, the gamete or the sex cell will contain the dominant allele, and in half the case, it will contain the recessive allele. And then it's just a matter of genetic lottery in terms of which sperm cell from the male and which egg cell from the female will actually fertilize. And so the Punnett square basically shows a sort of a mathematical expectation of what will happen in the future. And so in this case, the main point is that the sex cells contain a single allele, and that any one egg cell can fertilize any other sperm cell, and then that will produce the diploid offspring. His second law was the law of independent assortment. So basically, if you're looking at two traits, so instead of just looking at flower color, if you're looking at flower color and, say, the shape of the seed or the color of the pod or how tall the plant is, so any two plant characteristics, if you track those from parent to offspring and from offspring, which is the F1 to the F2 generation, each of those traits can separate. So you can have two traits. So one trait would be yellow pea pods and green pea pods, green being dominant. And in the other case, you could have a tall plant and a short plant. And so you could figure out all the different combinations that can exist from there. So you can have a tall green, you could have a tall yellow, you could have a short green and have a short yellow. And so this is what we call a dihybrid cross. So we're looking at two traits sort of going from parent to offspring together. And what he found was that for most of those traits, the traits do transmit or move separately. The only exception to that rule would be if the genes for those two traits are actually on the same chromosome. And so since chromosomes are transmitted through the sex cells, if the genes are on the same chromosome, then we say that they're linked, and so therefore they don't separate. So now the question is, what is the nature of the genetic material that genetics is based upon? So second big idea is that gametes or sex cells have half the genetic material of other body cells. So somatic cells are 2N or diploid, whereas the sex cells are haploid, having one copy of each chromosome. So if you look at the compare-contrast between the diploid cells on the left and the haploid cells on the right, you'll notice that they are essentially the same, except for diploid cells have double of each of the numbers. Each of these is a chromosome. Each chromosome numbered 1 through 23 is unique. The 23rd pair is different than the first 22 in that it can have an X and an X, as you see pictured here, or an X and a Y, but essentially they are duplicates of each other. In the left-hand side, we have one of each of the 22 copies from mom, one from dad, and then each of the parents donates the sex chromosomes. So in this left-hand case, if we looked at this as a somatic cell, or at least a non-sex cell, this is what would be found inside the nucleus. And in this right-hand side, we have a sperm cell that's penetrating the cell membrane of the egg. And so once that happens, then fertilization occurs. But prior to that, you have two cells that are both haploid, and when they fuse together, then they will become diploid. Big idea number 3 is that most of the cells in a human contain 46 chromosomes, 2 copies of each of these 22 autosomes, so that makes 44 total, and then another pair of sex chromosomes that determines the sex of the individual. And so 22 pairs of autosomes, which are 44, plus another pair of sex chromosomes gives us that 46 number. So if we look at the way that the cell is structured, how is it built, you'll see that on the largest level, the biggest scale, we have a cell. If we broke open that cell and sorted through the organelles, we would see, among other things, there's the rough and smooth ER and the Golgi body, mitochondria. If you're talking about a plant, then it also include the chloroplast, but you also find the nucleus. And so the nucleus is often considered to be the control center or the headquarters of the cell. And if we broke open that nucleus, we'd find chromosomes. And if we sort of zoomed in on one of the arms of the chromosome, we would find these what are called genes, which are long stretches of DNA letters that code for specific traits. So when you think about gene for eye color or hair texture or skin tone, those traits actually get tracked down to the very DNA level. And then the gene is made of nucleotides, in this case adenine, thymine, cytosine, and guanine. So if we look at the sort of structure of the nucleus, we'll see that there are these chromosomes. The diploid number for humans is 46. Every species has its unique diploid number, and then when that species is sexually reproducing, they also have haploid cells, which are sex cells. And not all organisms will reproduce sexually, but sexual reproduction is the best way to create a great number of different kinds of organisms because of just the randomness of things like crossing over and independent assortment and random fertilization that happen between one generation and the next. The last big idea that we're going to go over is that an organism's genes determine what traits it has. These traits can be dominant or recessive depending on the alleles found on their genes. So there's 4 hereditary patterns that you should become familiar with. The first is complete dominance. And complete dominance is defined by 3 different genotypes and 2 different phenotypes. So in this case, whether or not you are homozygous dominant or heterozygous, you're still going to look the dominant part, and it's because of the single dominant allele that's present. The only way that you can be recessive is to have 2 recessive alleles that pair together. So in this case, the dominant allele will always express itself over the recessive allele, and so that's why only when the recessives pair the 2 recessive alleles together can they actually get the word out, or they can express what their genes are showing. Here's a list of some common traits that are coded for by complete dominance. Likely you'll have a number of these different traits if you look at not only yourself but also members of your family. The second hereditary pattern is incomplete dominance, and this is a little different than the first one because there are 3 different phenotypes for 3 different genotypes. So no longer is the homozygous dominant and the heterozygous coded for as dominant. So it's easier to figure out, okay, well, which is the heterozygote. It will look distinctly different. And so in this case, there is no dominant phenotype. The dominant allele doesn't necessarily win over the recessive allele in the heterozygote. And so you have 3 different phenotypes and 3 different genotypes. A couple examples that come up again and again and again with incomplete dominance is cow color and flower color. So let's start with the flower color. A common flower in gardens would be the snapdragon, and in this case they have 3 different shades of color. One is red, one is pink, and one is white. The genotypes of each of these is different. So big R, little r is not red as if it were in complete dominance, and so that's a big difference for you to sort out. And in this case, if we have eggs produced by a heterozygote, which would be pink, and if we have another pink flower producing the sperm, which is the pollen, and they mated together, then 50% of their offspring, or 2 out of 4, would be the heterozygous pink, whereas only 25% would be either red or white. For cows, this is kind of how that works. The way that we write the genotype is a little different. Instead of using uppercase and lowercase, they actually use different letters entirely. So in this case, the full recessive would be the white, which is capital W, capital W. The full dominant genotype would be big R, big R. And then when they cross and have offspring, they have this heterozygote called roan. And roan is, as you can see in this picture, kind of a muted red. So it's almost like you took the white color and you just sort of faded out the intense rust color of the steer. Codominance is another example of hereditary patterns. The most common example would be the ABO blood groups. And so in this case, there's not really any competition between the different traits. They're both expressed together, so they have no qualms, no issues with each other. They both can sort of peaceably get along. So in my case, I'm A positive. Blood types are usually a letter and either a negative or a positive. So we're not going to deal with the negatives or positives right now. That's called the rhesus factor. We'll look basically at the letters. So if you say that you're type A blood, that could mean, genetically speaking, that you have this I with the A attached in the superscript or upstairs position. So you could be two pairs of those A's, or you can only have a single copy of the A as you see here, and then the second allele is basically blank. It's a recessive. And so basically, if you have type A blood, your body is going to reject anything that doesn't look like you, and so your body is going to make antibodies or molecules that are going to attack non-A blood cells. And so any B blood in your body will actually cause an immune reaction, was what we see in these last two columns. If you're a type B blood, that means that you have two copies of the B gene or just a single copy, whereas this O is really not impacting the phenotype, and that you're going to react against the A blood. And so this is why blood typing in blood banks and when transfusions are done in hospitals is very, very important that they actually get the correct blood type of the recipient, because if they make a mistake there, that could end up killing the patient. Type AB blood is actually the universal recipient. So AB blood can receive A blood or B blood or even O blood because it has the A allele and the B allele. So it doesn't have a very sensitive immune system. And then type O blood is called the universal donor. And since, in effect, the blood cell is naked, it doesn't have any protein attached to it, any blood protein, that there's nothing to sort of raise the alarms of any recipient. So if you're type A, type B, or type AB, you can receive blood from that individual. And so they are very, very, I guess, important for blood banks. They always want to keep a lot of O blood on hand, because if they have a major accident or some sort of casualty that maybe they need, they don't have time to type the blood, they can just give them O blood and everything will be fine. So in this case, A's and B's can sort of be expressed at the same time without any complications, so they're codominant. The last one would be sex-linked, and sex-linked traits appear really more often in either males if it's a X-linked trait, or females if it's certain X-linked traits. So let's take a look at what that would look like. So not all chromosomes are alike. We have the last pair of chromosomes, the X chromosome and the Y chromosome. You'll notice that between the two of them, the Y chromosome is much smaller, so there's much less genetic real estate on the Y chromosome. But even so, it has some important genes, one of which determines gender. The X chromosome is much larger and actually has quite a few important genes on it. In addition to those, you have pattern baldness, which can pass from mother to son, and you have hemophilia, which is an inability to stop bleeding. So if you get a bruise or a cut, generally your body will form a clot that would stop that bleeding. And in this case, it's a genetic disorder, so you're born with it. And if you can't stop the bleeding, then you can definitely bleed to death. So it's pretty serious. And then color blindness would be an inability to see red or green or variations of those colors. So it's the Y chromosome that makes boys boys, and it's the absence of the Y chromosome which makes girls girls. So XX is the sex chromosomes that determine females in most cases. There's some exceptions, but in most cases it's the girl combination, and then XY is boy. Pattern baldness is found on the X chromosome, so it's called X-linked. Whether it's found on the Y chromosome or whether it's found on the X chromosome, either of them together would be they're called sex-linked. This is a fairly large family tree from back in the 18th century, 19th century. This is the English monarchy, going back to Edward, the Duke of Kent, and Victoria, who was his wife. And you'll notice that we have circles and squares. Circles are female and squares are male. Carriers who are not affected but have the ability to transmit that gene to their offspring are noted in the half-shaded figure. So in this case, you'll notice that there are looks like 6 different women in this large family that were carriers, but none of them actually had it. So no females in this family tree are fully shaded in. So none of them had a hemophilia. It's only the sons in these cases. All of the red squares are indicating these sons, and modern medicine has helped a great deal to reduce the number of deaths from hemophilia, but 200 years ago, 300 years ago, these children generally died in young childhood as they play around and get injured and end up they can't stop that bleeding. So how does this affect things? Basically, if you're a male, 1 strike, you're out. You have a bad X chromosome, that's all you have, so you have the disease. For females, 2 strikes and you're out. So in most of the cases, it's the males that are affected more greatly. So they are suffering to a greater degree than the females, because females have that second X chromosome that is kind of their backup. So you'll notice in this case, the mother in this parental unit, she's a carrier, and so she gives X chromosomes to all of her children, and it's the Y chromosome that comes from the dad. And so the first and the third children are both sons, and the first son lucked out. He got the good gene, the good X from mom, the black X, and the second son was unfortunate, and he got the recessive X chromosome, and so that's his only copy, so he has hemophilia as a result. In this case, the daughters, they can get the genes from their mom, but they need a second copy to have the disease, and so that would have to come from dad. So if dad is hemophilia and mom's a carrier, then the daughters are definitely in danger. So you can see 2 different scenarios. One on the left would be the dad with hemophilia, and on the right, the mom would be the carrier. And so if the dad has hemophilia, the daughter can be a carrier, but only when the mother is either a carrier or has a hemophilia would the daughters be at great risk. The last example of X-linked traits would be color blindness. And so in this sort of collage, you'll see those 4 numbers. Anyone who is colorblind in red or green or other variations, they would not see all of these numbers. So from upper left, there is a 58. Upper right is an 18. Lower left would be a 2. And then the lower right would be a 17. So this would be a quick way to know if you are colorblind. If you have a variation of color blindness, then those colors would be kind of a grayish color. So that ends this podcast. So don't forget to write down your thoughts and questions and any ahas that you have. Thanks so much for joining in. In this podcast, you saw how Gregor Mendel was the father of genetics. He experimented on pea plants and worked out his 2 laws of heredity, which then paved the way for other genetic scientists to explain heredity. Then you were shown that chromosomes are the basic unit of heredity that are passed down in sex cells, and that these chromosomes are made of DNA, and that the gene is a string of DNA letters that code for all of an organism's traits. And then lastly, you saw the 4 main patterns of heredity, and that they were explained as follows: complete dominance, incomplete dominance, codominance, and sex-linked traits. So thanks for joining, and I hope this has been helpful for you. See you next time.